Hi everyone, thanks for joining this session remotely. My name is Becca Zombeck, and today I'll be sharing with you my research on how we may be able to use cool communities called biological soil crusts as analogs for some of Earth's oldest terrestrial life. So why do we care about this old terrestrial life anyway? What's so cool about it? Well, aside from the just sort of curiosity fact of finding life on Earth over 2 billion years old, there's a really good chance that when life did evolve on land, it would have affected terrestrial nutrient cycles and continental weathering. And both of those have implications for marine productivity and the drawdown of CO2 and the accumulation of oxygen in the atmosphere. As these mats form on land, they very well could have been cycling as these mats began to accumulate on land, they could have been cycling carbon and oxygen through photosynthesis, as well as releasing methane. Like today, they probably could have been cycling nitrogen by fixing nitrogen from the atmosphere. And then they also cycle, they could also be cycling an array of major and micronutrients ranging from carbon and phosphorus to iron and nickel and manganese and things like that. And so the motivation for finding evidence of this early terrestrial life goes beyond just knowing when and why and where life evolved on land, which is cool in of itself. But it has broader implications for marine productivity and how the atmosphere changed through time. And we do know that there were terrestrial communities a long time ago. We have some fossil evidence. Here we have some vesicles that were probably filled with gas, like we have in its modern counterpart on the right. But we can't only rely on physical evidence for reconstructing early life on land, because some abiotic processes could create structures that look similar in the rock record. And so we can pull in geochemical evidence as well. And so by looking at the geochemistry of these rocks, where we also see these physical structures, we can try and get a fingerprint from the microbial life. So it was saying, aha, like we were here, this is what we were doing. Which is not as easy as it may sound, since these rocks are, well over 2 billion years old and they've been put through the ringer. So while ideally we might find something like a carbon or nitrogen isotope that's very clearly reflecting, the carbon or nitrogen cycle, often what's left behind is more like a relative accumulation or depletion of elements and metals that may have been cycled by these microbial systems. But even if we can find these geochemical traces in a rock that we think has these physical microbial structures, our ability to interpret this geochemical signature is only as good as our understanding of their modern counterparts. And that is where these analog systems come in. An analog environment in the modern is something that a modern analog environment is a community that we think contains similar organisms and does similar metabolisms as their ancient counterpart, or at least similar enough that we can study them to try and learn about how their ancient counterparts may have behaved, what they were cycling, and importantly, what geochemical signatures they may have left behind in the sediment. And because a lot of research in ancient life has focused in sort of aquatic and marine systems, a lot of the analog systems that we're studying today are also in those environments. So on the left, we have two images from an analog community that a former lab mate of mine, Katie Rico, was working on. Pretty cool / creepy. On the right, stromatolites from Shark Bay, very clearly similar to the stromatolites that we find in the rock record. But we do also have some potential analogs for some very understudied early dry land ecosystems. and these are biological soil crusts. Biological soil crusts are symbiotic communities of bacteria, lichens, and mosses that form in arid environments, either where there's no sort of larger vegetation or between this larger vegetation. And they serve as hotspots for nutrient cycling and carbon accumulation, and they can hold water and do a really good job of stabilizing landscape. So they're really important in just a modern ecosystem sense, in addition to this interesting use of them as an analog. And so because, what am I saying? Yeah. And so we think they're pretty similar to the early microbial mats that could have lived on land, especially the ones that are dominated by cyanobacteria and haven't yet been colonized by some of the higher species like mosses and lichens. And so our questions in using these soil crust communities as analogs are really focused around how can we constrain what they're doing today, and how can we use that to interpret what we see in the past, and also just how realistic is their use, how reasonable is it. And so this first one is, do we see clear links between the microbial community and the potential biosignatures that we see in the sediment underneath? Secondly, do we see within the soil crust changes in redox microenvironments that we can link to relative enrichments or depletions of metals and other trace elements? And taking this genetic and geochemical data together, what do we think the possibilities are with this analog system, and what are the limitations? How can we most appropriately apply this tool? And so to explore these questions, my undergrad, Katie here on the left, and I headed out to Moab, Utah back in June. And we spent a couple of days sampling these really big, really well-established microbial communities outside of Canyonlands National Park. And so if you look on the ground here, all of this sort of bumpy, dark land, that is covered by soil crusts. And so these are systems that are pretty fragile. Even just walking on them can destroy them, and they take tens of years to reestablish. So if you're walking out in the desert and see a little sign that says "Don't bust the crust," that's what this is. That's what the sign is referring to. And so when we were sampling, we tried to capture an array of the different sort of morphologies that you can sort of see here. We have this sort of bumpy texture that's lighter in color, and then a little bit smoother but darker over here, and also a little bit of the very slight slope that we see here. So we really wanted to capture as much variability in these ecosystems as we could. And a lot of the variability that we were interested in exploring is specifically related to their ecological succession. So on the left, we have a pretty young, immature, or poorly established soil crust. You can see it's pretty thin, just 1 or 2 millimeters. And if you look, you can actually see these filaments sticking out. And so this is the cyanobacteria just really starting to aggregate the soil together and form the very beginning of a crust. And then as it becomes more established, you can start to see the sort of patchy texture. Even just pressing a finger into this will leave a dent, and you can see the crust sort of holding up around it. And then in these later successions, we see more mounds or pinnacles that start to be darker in color. And finally, we see lichens and mosses start to join in. And then this is sort of your final stage. And these numbers 1 through 4 just refer to this succession, and I'll be referring back to those numbers throughout the talk. So just for your reference, just remember 1 is sort of immature, and 4 is relatively mature and darker in color. So just briefly to do this work, we did a 16S analysis sequenced here at Michigan. And we did an interesting application of a sequential iron extraction. It's actually novel. People haven't applied it to soil crusts before, so that was interesting. And then finally, to get at our sort of elemental analysis and the concentrations that we would use for biosignatures, we just did a bulk analysis of the crust and the underlying sediments, and sediments that did not appear to have crusts. And so just to get into our community analysis. These are just the top 10 phyla. It's pretty big umbrella to be under. Acidobacteria are big players. We did actually have some issues in getting breeds of our cyanobacteria to behave, and so here they're just lumped in to the unclassifieds because the postdoc I was working with had to use a separate reference library to get them to pull out an older one. So I've just included them here because there's a little bit of uncertainty about what the actual abundance for those is. If anything, it's a little bit low because we do know that cyanobacteria are sort of the backbone of these communities. But the big takeaway here is just that the community that I found is quite similar to other community analyses that have been done on biological soil crusts, and particularly actually around this region. So even though they are quite varied in terms of the metabolisms or redox environments that they would be indicating, we at least have consistency in that they're highly they're highly variable. And then in terms of diversity, alpha diversity was quite invariable, so I didn't include a figure on it here. But beta diversity showed a little bit of a pattern based on soil morphology. And so overall, they're quite dissimilar if we're comparing between a light crust and a dark crust, or that is an immature and a mature crust. Where we see the least dissimilarity, or the most similarity, is actually between dark crusts, so these well-established ones. And so even though they're still quite different, maybe they've been around for a while, they've really honed in who they need to have around, and so they're a little bit more similar there. But the big takeaways here are just essentially, again, that these communities are quite complex and basically just really hard to predict, which isn't a great indicator if you're looking for a potential analog community. So we have all of these different phyla, they're doing all of these different things and suggesting all these different sort of redox microenvironments, and so that sort of throws our nice hypothesis about crusts holding on to water and cycling nutrients in specific ways sort of out the door. But all hope is not lost because in addition to these genomic data, we do have that geochemical data that we can now turn to try and get a more nuanced idea of what's going on here. And so first I want to talk about the redox microenvironments. And so just as a reminder, here we're looking at comparing the crusts that are coherent. You can actually pull them up and they stick together. That's compared to just sand that does not appear to have any microbial community on it. Apologize for the sort of bad quality of the sand. I was out here in Utah taking all these great pictures of these cool crusts, and I forgot to take a good picture of just plain sand because who needs to see that? And so our expectations had been that we would see an enrichment of the sort of poorly crystalline reduced iron in the crust compared to the underlying sand because of that water holding capability. However, we saw the opposite. So the crusts were depleted in this labile iron and actually enriched in iron oxides. And we found no difference in iron carbonates between the two parts either. And so the big takeaway here is that despite the water holding capability of the crusts, it actually seems like once they're wet, they dry out really quickly and just stay dry and stay oxidizing, which is actually a big plus in thinking about this as an analog community and thinking about the implications for sort of early Earth, because the ratio of reducing to oxidizing conditions in these fossilized soils has actually been used as a way to think about how much oxygen was in the atmosphere. And so this is just a nice little check that these soil crusts, even though they can be reducing sort of short-term, are long-term recording oxidizing conditions. So that's a good check. And then also, we don't really see any trends in how well established the crust is between redox state, which is sort of surprising because we would expect that something that has mosses and lichens would be much better at holding onto that water for a longer period of time. But the changes in redox condition are probably just so transient following a precipitation event that it's just there's no really there's no real trace of those reducing conditions left behind in terms of the iron geochemistry. And then finally, sort of the meat of what we're getting at is what elements could we maybe use for biosignatures. And so here again, we just have some major and sort of minor elements plotted out by crust type. We didn't have any bulk geochemistry for the very incipient soils, which is unfortunate. Next time, though. And so the big things here are that we don't really see any strong trends between geochemical enrichment or depletion and maturity. Maybe some, like we're looking here at copper, potentially phosphorus, but overall it seems like things are either depleted or enriched. And even within soil morphologies, we have some inconsistent or mixed results. And so what this really points to is a need for more information, need for more samples, like almost everything in geology. The good news is we did see some agreement with previous biosignature work. So calcium, manganese, and arsenic were all depleted in our work, with the exception of this one sort of weird sample in terms of calcium content. And so this agrees with previous work. There is some disagreement. So these three elements, copper, chromium, and zircon, were all depleted in previous work, whereas here we see some enrichment, some mixed results. And so again, this points to just a need for more repeats of these sorts of studies so we can start to figure out which elements are consistently and significantly depleted or enriched relative to the underlying substrate sand. And then we also see some trends in terms of what is consistently depleted. So for example, iron, they didn't look at, but we found to be depleted. Manganese is depleted, and nickel is also depleted. And so to me, this seems like a pretty clear indication of redox-sensitive elements being leached from the crust, mobilized during these precipitation events, and then deposited essentially as a biosignature below the sediment. So our big takeaways here are just that we have a couple of potential biosignatures. We would still probably want to do some more work to really establish them. And then there is still inconsistency between studies and even within an individual study in terms of just repeatability of your results for what's enriched versus what's depleted. And it all circles back to just the fact that these are very complex systems that can change at a moment's notice depending on how much it's rained recently. So our big takeaways from all of this work are threefold. So the first one is that just community level analysis like this is really not nuanced enough to pair with geochemistry. We really should have something like transcriptomics, so that we can see which genes are being activated, which metabolisms are really active, not just what is there and what has the potential to be active, and what could leave a biosignature. Secondly, it does seem like these crusts could be leaving some biosignatures, but it may be variable depending on where your crust is, what stage in sort of the precipitation drying out cycle it is. And so more work is needed to determine which signatures could be robust sort of across different studies and different soil crust sites. And finally, in terms of what this means for an analog and our ability to interpret our results in that sense, an important sort of caveat to all of this in a biosignature sense is the fact that this is done under a modern, highly oxidizing atmosphere. And so while it's good to understand how that works, and it's applicable after, say, 2.1 billion years ago, after oxygen really rose in the atmosphere, if we want to think about these sorts of systems prior to that, we really need some like growth chamber style experiments where we can control the amount of oxygen and carbon dioxide in the atmosphere to see how metal mobilization and nutrient cycling changes under those conditions and how the biosignatures change in that sense. So with that, just to wrap up, I like to thank funding sources here. the USGS Moab office, and Jane Belknap, the Soil Crust Queen, for her very helpful walk among the crusts. And finally, I want to congratulate my undergrad, Katie, who graduated in the midst of coronavirus and was just a real trooper in getting her work with the iron redox stuff done, and being really flexible in how she ended up doing her thesis. And I look forward to talking with you all about this.